Thank you very much. I never knew Denver Russell because he was a bacteriologist and I'm not. But there are people at NIBSC who did know him, and I think we liked him quite a lot. And I'm really quite honoured and pleased to be here to actually give this particular lecture. Okay. What I'm really going to be talking about is the end bits of polio, okay? And it's really not a straightforward business at all, okay? It's been a heroic effort. This is the largest single public health intervention against a single disease entity in the history of the human race, and believe me, it shows. Actually, it's really most impressive stuff, okay? But before I start, I wanted to talk a little bit about other eradication programs. This is just one slide on this, actually. Smallpox has been eradicated, rinderpest has been eradicated. There are things maybe that you think you might learn from those, perhaps. Guinea worm is Jimmy Carter's pet eradication program. He took that over when he stopped being President of the United States. And that is a really wonderful program because it's very appropriate technology driven and so on and so forth. And of course, polio is very nearly there. My view of can you learn anything from any other eradication program is no, you cannot. It depends on the disease you're looking at. The only thing you can learn is that one, you must get rid of the agent, never mind the disease. And number two, you're going to be short of money. Okay. I've never read anything on these things where money has not been the big deal. If you read the smallpox stuff, it's always money. So there are serious comparisons made currently with the end game about polio and smallpox, and I think they are spurious and they are actually causing, I think, a bit of confusion. You must not misunderstand anything that I say to be a criticism of anything to do with the programme. I think this is absolutely fantastic, okay? But nonetheless, I'm going to be critical of the programme. So I divided this into about 5 different parts. There's prehistory, which is the background to the whole polio business. There's an awful lot of background in polio, and I'm going to skate over it to some extent anyway. Okay. I'll then talk about the wild virus and where that's actually at in terms of eradication, which is very close indeed. I'll talk about oral polio vaccine, which is beginning to cause the problems which we are now having to deal with. I'll talk about the withdrawal of type 2 from the oral polio vaccine, which has implications which are not very good for anybody in this room, I suspect. Okay, and finally the implications of that at the end. Okay, and at the end of it, I will maybe summarise a little bit. So prehistory. Now, I always thought Copenhagen was a very boring place. Okay, but we went there for some scientific meeting, and it really isn't. If you ever go there, make sure you do the brewery tour because it's fascinating. There's all human life is there. Jacob Carlsberg was the paterfamilias who started the brewery. Okay? His son was called Carl Jacobsen, okay, in the manner of Scandinavians. They hated each other, okay, and the story of the dynamic between the two is incredible. The younger lad collected antiquities wherever he went because they were both filthy rich, and in the end he built a museum to put them all in because he had so many of them, and it's called the Carlsberg New Glyptotek. And if you come out of Copenhagen main station, hang a right, that's that big building about 100 yards down the road in front of you. And if you get a chance to see it, because it's not just this that's wonderful in there, there's a lot of other wonderful stuff too. This is the funerary stele of the priest Rom, or Roma. And it's about 9 inches tall and about 3 inches wide, okay? And they don't make a big thing out of it, but this is the most famous funerary stele in the whole of virology. This is a guy who clearly has polio. If you look at his right leg, you see that it's withered. There's actually no musculature there at all. Just in case you think that's because the man couldn't draw, it's not true. Look, he's got the downflex foot. There is no muscle tone in his leg whatsoever, okay? And he's got the stake that means he walks, okay? This man has no motor innervation to his right leg, basically. And the easiest way to fix that is to get him have polio, okay? That's generally speaking the accepted view that Roma actually had polio. It's not, it could be something else, but I don't think so. Right? And that's from 1400 BC, so it's late Middle Kingdom Egyptian, early New Kingdom Egyptian, and they make nothing of it at all. It's just this tiny little grey thing down here saying this is the funerary stele of the priest Roma. They don't say anything about how wonderful this is. Whole books have been written about Egyptian medicine based on this one artifact alone, and I'm not kidding. Right? So what happened after that was basically nothing. Okay? Now, bearing in mind that the clinical presentation of this disease is so absolutely characteristic that you can diagnose it by hieroglyphics, you'd have thought if it happened afterwards, really you would see it in the literature. And there really isn't very much at all. There's a handful in the 18th century, there's another handful in the 19th century, okay? But generally speaking, there wasn't any polio to speak of. And that's quite strange, I think. So until the end of the mid to late 19th century, no polio at all. Then at the end of the 19th century, it began to emerge in epidemic waves, which would happen in the summer. It began in Sweden, and by about 1909 or thereabouts, it turned up in America. Okay? And that's when things really began to go seriously interesting in general outline and shape. The Swedes, in my opinion, more or less had it taped by about 1912, 1909 or thereabouts. Okay? However, throughout the 20th century, there were really terrifying epidemics of polio every year in the summer, mostly in the first half of the 20th century, but throughout, really. And what would happen would be that in the summer you get the epidemic and then it would go away and then you get the epidemic again. There's a lot of work was done on this, a lot of quite misleading stuff done with animal models, particularly monkeys, and that's one of the things that the animal rights guys come back on occasionally from time to time. But by the time you got to the end of the 1950s, certainly in this country, polio had really disappeared, actually. And if you look at the figures for polio from the Department of Health, and it's on an arithmetic scale, you can see that really this was a disease of the 1950s in this country, not much before and not much after. So by about 1960, 1970-ish, polio had really been controlled in developed countries like here, most of Europe and the United States. But at that time, it was really thought that the vaccines did not work well in tropical countries. Okay, and there are a couple of reasons why that would have been the case. Okay, so while vaccine was being poured into them, they were not really doing much about making a dent in the incidence of polio as it was occurring. That changed in around 1985 or thereabouts when Sabin got involved in a South American effort. And it began to look as though you could actually wipe out polio from somewhere like South America, which is a middle income, if you like, but nonetheless it's not like North America. And WHO picked up on this and said, then we can eradicate it, let's do that. Okay, so that was the phase 3, if you like. Okay. Phase 4 is getting rid of the vaccine. The vaccine that was used almost exclusively in this particular phase was oral polio vaccine, which infects you without causing disease. But it's a live virus and it goes into your gut and it will stay there for 30 days at least. In my children, it was more like 50 and 70, things like that, quite a long time, when there's plenty of time for the virus to do whatever it needs to do to make itself happy. And we'll talk about that in a minute. This actually causes big problems with the getting rid of it. And you have to get rid of the vaccine to get rid of the disease as well. So that where we are at the moment, really, is about stage 3.5. Okay, there's a handful of cases left in the world. And the vaccine is basically on the verge of steps are being taken to actually change how we do it. Okay. Those of you who are younger than I am will find pathogenesis really, really boring. But believe me, as you get older, pathogenesis gets to be more and more it. Okay. Some people don't live long enough to find pathogenesis interesting. Well, stick with it, guys, because this will do it for you. This is according to Sabin. There's another version of this which is according to Bodian, which is oversimplified. And this is actually oversimplified as well. I mean, it looks impossible, but it's oversimplified. In a word, and this is essential to the actual eradication program, this particular pathogenesis, where the problems come from. The ingested virus comes in at the top, and it's basically just stuff contaminated with human excrement, basically. Okay. It'll then either go to the oropharyngeal mucosa, so it can infect your tonsils or get shed from your throat. More likely, it doesn't do that. More likely, it will go into your intestinal mucosa somewhere. Nobody really knows what cells it grows in or where it grows in the intestinal mucosa, but that's where it grows. Okay? And if it stays there, there's no issue. Okay? The virus will get shed in the feces for a long time, or as long as it takes. Okay? And it won't do any damage at all. And in fact, you won't know you've actually got it. And that's the key feature of polio. Okay? Most infections are already completely silent. The virus can leak out into the Peyer's patches, the mesenteric lymph nodes, and then you get a small viremia, which will seed other things. Okay, so like other susceptible extraneural tissues. This is fairly gnomic, but it happens. Okay, and as a result of that, you get a secondary viremia, which comes back and begins to spread things around much more. So as a result of these viremias, you can get your oropharyngeal mucosa infected, and you can also get the regional nerve ganglia infected, and you can get your central nervous system infected. If you can stop that bit, then you've basically stopped polio-causing disease. Okay? And it was shown in the 1950s that humoral immunity, so passive immunoglobulin, would actually prevent polio occurring. Okay? This is a study by Hammond in about 1955 or thereabouts. And if you can do that, you can confine it to the gut, there is no issue. Okay? In fact, you don't know you've got the disease or the infection. This is work by Wickman from about 1905. He was an epidemiologist avant la lettre. And he committed suicide in the end because he didn't get to be a professor. So just stay loose, guys. It's not that bad, believe me. And what he showed was, investigating an outbreak about who'd talked to who and who shook hands with who and what was going on, was that there were really three clear phases to the whole process of getting polio. Okay? Asymptomatic infection. He showed that 99% of them were actually completely asymptomatic. You could not tell that somebody had this, okay, at that time at all, by any means. Okay? There's a thing called the minor disease, which is like a systemic infection by a virus. It's like malaise, sore throat, headache, that sort of thing, but nothing particularly serious. 3 to 4 days post-infection. And then later on you get the major disease, which is what we call poliomyelitis. There can be aseptic meningitis, and we won't talk about that. That's just non-paralytic polio. Again, it affects the brain a bit. The other stuff is really what everybody calls poliomyelitis. So you get spinal polio, which is what Rom had, down in the legs. You can get bulbar poliomyelitis, bulb being an old word for the respiratory centre bit, okay? And that's where all the iron lungs came from in the 1950s because of those kind of cases, if you like. Very serious. And you can also get encephalitis. The key feature of polio is it goes for motor neurons, not sensory neurons, for what it's worth. Okay? So what you find is that most infections are silent. At most, 1% of those are probably going to go and lead on to the major disease. So it's very difficult to know what you're doing. Okay? I will clarify that in a moment. This shows the incidence of polio in the United States from about 1950 to about 1980. Okay? So you start off at a level of about 10 cases per 100,000, and that is common in a developed country without any vaccination whatsoever. Okay? So it was very similar over here. We had about 4,000 cases a year on average, depending on was it an epidemic or not. Okay? The inactivated vaccine, which is Salk's thing, which is polio virus which had been treated with formalin very carefully to keep it functional, that is to say antigenically functional, was introduced in the mid-50s. There were certain issues with that, which we won't go into. But by the time the oral vaccine was introduced in 1962, the rate had been reduced by about 95%. And I suspect if they'd carried on just using the Salk vaccine, they could have handled it completely. Okay? We used it here as well. Okay? And by the end of the 60s, we'd really got rid of most of it. But the oral vaccine was introduced, and there was always an ongoing fight between the virologists who thought you could prevent disease by using an activated virus, and those who thought you had to have a live virus to make sure you get all the kind of immune responses you need. But it was introduced in 1962, and that was Sabin's contribution. And it carried on going down there, and finally it bobbled around at that sort of little rate that you see there now. Okay? And there was a fierce argument about this at the time, about was this to do with the vaccine or was it wild-type viruses that we were missing? And frankly, I think secretly everybody knew it was the vaccine. The first case of vaccine-associated poliomyelitis that I know in this country was in 1962, and the vaccine wasn't introduced till 1963. So I think that tells you that people had a pretty good idea of what was going on before that. And we'll get back to that in section 3 or so. Okay? But you can clearly see that in developed countries, it was controlled quite easily. There are 2 vaccine types, as I've said, the inactivated polio vaccine, formalin-treated poliovirus, which is developed by Jonas Salk. There's a far more sophisticated and better version of this now using purified virus. It's much, much more potent and much easier to control. And the oral polio vaccine, which Sabin used, which was a live attenuated strain. And it's the OPV, which is the backbone of the eradication program, or has been so far. Right, so here are the scientific difficulties of polio eradication, and they all arise from the polio pathogenesis that I just described. Okay? Most infections are silent. Okay? Therefore, there is no simple way to say if someone is infected or infectious. Okay? You cannot put people in quarantine in a polio epidemic, and people tried. I mean, they did it in New York in 1940 something or other. Completely pointless, because by the time you know you've got a polio going on there, all these people have been wandering around free as a bird, and they've been passing it on to everybody else. You cannot do it by quarantine. You can't do it by ring vaccination, which is what you could do in smallpox. Counting cases then is just a surrogate of saying, have you got it? Have you got infection in your population? And again, there's a specific example which I'll mention in a moment about that. It's just a surrogate, right? Okay. It's a pretty good surrogate if you've got massive cases, but it's just a surrogate nonetheless. The agent must be eradicating, not just the disease, and that's something which is in common for all eradication programs. If you don't get rid of the agent, you don't get rid of the disease, because sooner or later it's going to come leaking back. So that's a major message. You cannot just control polio, which is what people used to say. You must eradicate it, and you have to eradicate the agent. There's no way to tell if someone has been vaccinated without laboratory follow-up. It's by looking for antibodies. these are all very different from what happened in smallpox. So in smallpox, if you weren't openly festering full of sores and things, you were probably not infectious. I mean, to a first approximation. Okay, so you could tell where it was. You could also do ring vaccination and quarantine if you so wished. Okay. Counting cases was not just a surrogate of infection. It would a pretty accurate figure for infection. Again, the agent was eradicated. And there was also ways to tell if people are vaccinated as well, because you get this big scar on your arm. There's not with polio, you see. Okay. So what this means is then that the whole program is a much more heroic effort than it was with smallpox. And frankly, smallpox was pretty heroic as well, actually. But this is even bigger. Finally, in my pre-history bit, I want to do money. Okay. These are sort of health economics kind of considerations, and in my opinion, they are open to criticism. So the story with smallpox is that firstly, the eradication program was from 1967 to 1979, and it cost $306 million US. Okay? Now, I don't know if that's 1979 US dollars or 1967 US dollars. It's certainly the case that some countries had eradicated smallpox before that, like Bolivia, for example, had any cases for a long time. Okay? And it's not also clear to me whether that includes all the, what you might call non-financial inputs to it, like the infrastructural things. But that's the figure that people talk about. And it sounds dirt cheap, and it is, actually, but I don't know what that is in modern terms. Rinderpest finally lingered on in Africa. In many other parts of the world, it had gone, okay? And that cost $5 billion to get it out of Africa properly. So that's a major, major difference. Okay? Guinea worm is 155 million. I love this because it's such a local effort. I mean, the guinea worm goes under the skin, and you cut it and you wind it out on a stick over a period of weeks, you see, and then you make sure you don't get it again. There's no vaccine here. This is absolutely appropriate technology, and it's really, really neat. I love it, okay? And I think Jimmy Carter's a good guy, and I hope he gets to be president again. Polio, on the other hand, cost $11 billion from 1988 to 2013, okay? And the estimate at that time was that it would cost another $5.5 billion to finally get rid of it by 2018 proper. Okay, so this sounds like an awful lot of money, and frankly it is. It would certainly pay off my mortgage and probably yours as well. Okay? But I apologise for this stuff when I stand up and talk about it, say how expensive this is, and then regularly somebody comes up to me and says, how much is a stealth bomber? So there's the figure. Okay? Stealth bomber is £68.1 billion development costs. Unit cost 1.5 billion. You could eradicate polio then for 11 stealth bombers. Okay? The stealth fighter is what you might call the sense of this particular economic calculation. 66.7 billion to develop and 150 million to actually buy one. Okay, so you could have eradicated smallpox for 2 stealth fighters, and you can get rid of polio for 11 stealth bombers, and you could probably clear the world entirely of disease for however many, for the development cost that was actually involved here altogether. But as you know, people prefer to kill each other than actually cure disease. Right, so that's the introductory bit. Okay? What I'm now going to do is talk about the state of play with the wild-type virus, and I think this is tottering on the brink of eradication. So in 1988, okay, the WHO said, wouldn't it be a good idea to eradicate polio? And on this slide is the reason why they were able to actually say that. If you look at the top bit, 1988, it was estimated there were 350,000 cases, and there were probably a lot more than that because surveillance was basically rubbish at that time. And you can see that we haven't got it. France and Spain have it. They got it imported from Northern Africa. Africa's got it in spades. Everywhere else has got it in spades. Okay? South America, on the other hand, those 2 countries that my geography is dreadful, and I'm not going to even pretend that I know what countries they are, except I think one's Argentina, one's Chile, no, never mind, it doesn't matter. The 2 countries down the bottom, and Uruguay, the 2 countries down the bottom there had actually got rid of it, okay? And that was actually a new development, and that was Sabin doing his thing down there and saying, you must use my vaccine in this particular manner, and persuading them. Sabin was a very persuasive man, I believe. Yes, no, he was, I believe. And he showed that you can actually do this in a middle-income country. So WHO pounced on this and said, if we can do it there, we can do it anywhere. And they were right, okay? So the strategy then that was involved was mass campaigns, okay? And what we were doing over here at that time was we'd immunise the individual when they get to be a certain age. So it would be 2 months or 4 months or something like that, okay? So gradually they would get immunised as time went by, but it was depending on the patient, and that's the sort of thing that GPs like to do. They look after the patient, they don't look after the world. What you do here with getting rid of it in a developed developing type country is you swamp the entire country with a vaccine. Okay, so every child under the age of 5 gets a dose, basically. And what that effectively means is that suddenly your susceptible pool disappears because they've all been immunised altogether. Okay? So there's nowhere for the virus to go and it just dies out. The reason why it works in this country is that it's a very seasonal affair. So you get it in the summer, you carry on immunising through the winter, your susceptible pool gradually dwindles away to next to nothing. Okay? So it worked here, but it would not work in somewhere like Africa or somewhere like that. I think there's a second issue here as well, which is that the delivery of vaccines in this country, even back in the 50s, was not that awful. Okay? Whereas I think if you go somewhere hot with these kind of vaccines, it may very well be that they don't really last very well. And in a campaign, it's much easier to control that, I think. Okay. So anyway, that was the strategy, and it really, really worked like a charm. The position you're now in is this. So it's the red dots are type 1 poliomyelitis caused by wild type polio. Okay? And you've got it in Afghanistan, and you've got it in Pakistan, and you've got 4 cases in Nigeria there. Okay? At November last year, there have been 40 cases altogether in the world. Okay? That's down from however many million it was back in 1988. Okay? Ignore the purple spots and the green spots. I'll come back to those in a minute. The 4 spots in Nigeria that you see there were a big surprise. Nigeria had seen no polio at all for about 2 or 3 years, and it was just on the brink of being declared polio-free when suddenly 4 cases of polio appeared. When you look at the sequence of them, which is how you tell where they come from, you can show that they are actually Nigerian isolates. This stuff was circulating in Nigeria and nobody knew about it. Okay? And that's one of the big risks with polio, that you must keep looking to see if it's really, really seriously gone, and you can't really trust it before that. Okay? Okay, but in terms of where we're at, I think this is really desperately impressive to me. And the efforts that go into this are heroic, as I said. Here is an example of this particular heroism. The idea was that WHO were very worried about Nigeria because it was stiff with cases at the time. The program wasn't awfully well run there. Okay? And so what they thought they would do is they would concentrate their efforts on Nigeria and relax their efforts in the countries around it, okay? Which they duly did. At that moment, the people in the north of Nigeria decided that this was an American plot to sterilise their girl children. Okay? So basically vaccination stopped. Okay? It's a Western plot, if you like. Okay? So it stopped. And that would have been about 2002-03. What happened then was that polio leaked out of Nigeria across the whole of Central Africa there. You can see that. I'm no good at geography at all, as I said before, but you can see it's the whole of Central Africa basically has got it. Okay? Some of these cases were actually from viruses which had been circulating. The ones in Chad, for example, were Chad-specific viruses. Okay? So they had been circulating and nobody had noticed them, but the rest of them were Nigerian. And not only that, the Hajj came on, okay, and northern Nigeria is Muslim. They go on the Hajj, and the result of that is that it got put into Yemen, which is that big dollop of red spots there, okay, and also Indonesia down here, both of whom had eradicated polio previously. So this is interesting for 2 reasons. One, it says if one country in the world still has polio, the entire world is at risk. And in fact, what WHO say is that give it 10 years and you'd be back to 200,000 cases. I think it would be quicker than that. This was only a couple of years, and look at the number of cases there were there. So that's number 1. Number 2, the vaccination programs always target the little children under 5. Okay. Now, if you're on the Hajj, you're not a little child. Okay. I think you're an adult male, mostly. So that means the adult males also carry it, but it's probably not that significant in terms of infectious burden. There have been numerous examples of this kind of thing, but this is the thing which really emphasises it enormously, that if one country has polio, everybody's at risk. Okay. India used to export to South America, South Africa quite a lot on a regular basis. They also exported to Tajikistan on occasion, and so on. Okay? You must get rid of the virus if you're going to do anything about this. And I think that's almost where we are in terms of the wild virus. It's more or less gone. There has been no type 2 since 1999. There's been no type 3 since 2012. Those, I think, are eradicated. And I think somebody needs to say that, because you talk about the polio eradication program, you've eradicated 2/3 of it already. You see. However, oral polio vaccine becomes a problem. Okay. So this is a live vaccine which will infect the child or whoever it's given to. It will be excreted for a while. Polio is a very, very cunning virus, like all viruses. Don't tell me that polio vaccines are simple, mate. They're bloody not. Excuse me. So what happens is it goes in and it replicates and it adapts very, very quickly to conditions in the gut. Okay, and it really can play 52-card pickup with its genome and Lord knows what else. Okay, it recombines, it mutates, it goes on for a long time, and whatever obstruction you put in its way, I think it can probably wriggle out from under it. So some time ago then, there were meetings in WHO where they said, how can we ever stop vaccinating? Because this is one of the pluses, if you like, of an eradication program. And this was the apple pie and mother kind of statement that came out. It said vaccination with OPV may stop when wild-type virus is eradicated. That's number one. Okay, so you need to keep on looking to make sure it has been eradicated. If the vaccine either causes no disease or if it is unable to spread. Now, one of the big advantages of OPV in the early days was that it was thought to immunise spread to other people. It was you immunise one person and you catch another 3, and that was thought to be a plus. Okay, so you'll be not surprised to hear that it can spread. Okay, and it does. Not only that, but it also causes vaccine-associated poliomyelitis, or VAPPs. These are called. And this is the rate of about 1 in 750,000 primary vaccinees. Okay? So in this country, where you have a birth cohort of about 600,000 or so, you might have seen 1 a year. Okay? So very difficult to actually put a reasonable figure on what the rate actually is, but a meta-analysis of sorts was done which says that's basically the rate. So it can cause polio. Okay? It can also cause it in contacts, so that means it does spread as well. Okay? And it is believed that it can die out after about 3 person-to-person transmissions. And I think that's an average, right? If it goes on for long enough, I don't see any reason why it wouldn't regain the ability to spread from person to person. And of course, that's exactly what it does, okay? But this may be good enough, right? If you've got a low enough incidence of polio in the first place and a low enough incidence of transmission from one to the other, it may be that you're okay. You can get away with it. Roland Sutter, who's a big cheese in the polio program, said eradication programs are like rowing across Lake Geneva in a leaking boat. He says you find this big leak and then you nail something over it to stop it leaking, and then you paddle on a bit further and it leaks again and you patch up that one. What you hope is you get to the other side before it sinks. Okay, and I can see exactly what he means. Okay, so these are the problems with oral polio vaccine. There's the vaccine-associated paralytic poliomyelitis cases, which I just mentioned. That's in recipients or in their close contacts. Okay. So, well, we can get onto that in a minute. Circulating vaccine-derived polioviruses are CVDPVs, and they are caused by the virus actually adapting and becoming transmissible. There is some discussion, I think, about what this actually means. And what I think is going on is that you select for transmissibility of the virus, and after that it can do what it likes. Okay? If it wants to be virulent, it will be. Usually these things are about 2 years' worth of circulation, which you tell by sequence variation as it accumulates. Okay? So you can get these things, and they are very, I don't mean very common, but they are common. They're not rare. Okay, they've occurred in all sorts of places. Nigeria had a lot of them from the type 2 component. Okay. IVDPVs, these are immunodeficient vaccine-derived polioviruses, where you have a common variable immunodeficient, typically, so one who does not make humoral immunity, and they get given Sabin vaccine either accidentally or because they weren't diagnosed before it actually turned up. They can excrete for a long time, and I'll show you the world record holder who is British. We also have abnormal vaccine-derived polioviruses, okay? And these are things which just turn up, nobody knows where they've come from, okay? They may be just a sporadic isolate from a child, for example, who has flaccid paralysis or whatever, or they may be sewage samples, and it's the sewage samples which I think are the real issue. So I go back to this. So this is, where are we with the wild type? And the red dots are the type 1s, and the purple dots that you see in Lao there are type 1 vaccine-derived polioviruses. Okay? So they are sequenced, they are shown to be closely related to the vaccine strain that went in there. They're about 2 years' worth of circulation based on the genomic differences. And they're also recombinants too. They tend to be recombinants with other enterovirus Cs. They're not non-polio sort of thing. It's not clear why that is, but it's almost an invariable feature of it. And if you look over in Senegal on the left-hand side there, you see there's 2 little green dots, and that's type 2. Okay, and I think that's sort of really unfair. I mean, Senegal had just dealt with Ebola, and then they get polio. It's just not right, is it? Okay. So there are a few of these around now. And Nigeria had a lot of them, actually, which you can clear up by using monovalent oral polio vaccine, but we'll get back to that later. This happens, and it happens not uncommonly. This is an IV DPV, and this is work from my colleague Javier Martin at NIBSC. This man was discovered as a result of a study which was intended to find out why humoral immunodeficients have intestinal problems. Okay? So the idea was that you take one lot that have got diarrhea and gastroenteritis a lot and another lot which are your control group, and you see what pathogens are actually present. He was in the control group, so that supports the view earlier that most of your infections are entirely silent. He's meticulous in his immunoglobulin administration, and I suspect if he wasn't, he'd be dead, okay? I mean, because these are quite nasty viruses. It's type 2, and we have isolates here from 99 to 2000 up to the present date, about 2015, okay? And what we have here is just a lineage saying how genetically related these different isolates are. And you can see we've got L1 down there, which is out on the end, 99 to 2000. We've got L2, which is the second lineage, which is also present in 99 to 2000. And we've got L3, which is another one, 99 to 2000. You've got 3 separate foci of infection here, okay? They've been replicating independently for about 2 years, okay? So you've got one intestine, several sites of replication, and I don't understand how this works, because I was always brought up to think you've got an ecological niche, one should win out over the other, and it's not happening, okay? And it happens a lot. And you can get startling and sudden changes in the lineage which is predominating at any stage, okay? There's a big mystery about where polio grows and how it does it, actually, but these things are going on forever. And he's been going for now for about 30 years, and it's a nasty virus, and he's married, and we thought maybe we can actually cure him. He went to Thailand on his honeymoon, and we were hoping he was going to get some amoebic dysentery, which we thought might fix it, but he was very careful and he didn't. Okay. Shame. Okay. The only one of these that's actually been cured, by the way, got a Shigella infection. Okay. That reamed him out from head to toe. Right? And finally, there's the highly drifted VDPVs in sewage. So these are the abnormal or atypical VDPVs. Okay? Wherever anybody has looked in sewage, and believe me, polio virologists like to do that kind of thing, okay? They found these things. So Finland were very big on this. Okay? They found a couple there, a type 1 and a type 2. Slovakia, Estonia, Israel found them. Egypt had them for a while. It's also been turned up in Pakistan and other places, okay? Where you have a virus which is isolated in an environmental sample, okay, which is not related to any disease that you can detect, okay. Some of these at least are probably immunodeficients that you don't know about who are carrying on excreting, okay, but they are a bit of a mystery. In this country we have nowhere I don't think has actually got a decent immunodeficient registry, so you can't really go out and check them all, and that might be a worthwhile thing to do if you could bring yourself to do it. What we wanted to do was to do some environmental surveillance here. So, for example, there are certain loci in this country where you might expect to see polio imported or circulating or something like that. And there was certainly polio circulating in Israel who were using the same kind of immunization strategy that we use. So why wouldn't it happen here? So this has been set up with, we have colleagues in Pakistan that we're looking at. We've got colleagues in Glasgow, and PHE and us are working on a London, East London sewage catchment area, if you like. And we're turning up a load of different viruses. So these are all deep sequenced stuff, okay? And I think we wish to carry on doing that. You get a lot of enteroviruses coming through, which I think is a good positive control. It says that you're actually getting samples that are reasonable. And we got a poliovirus Sabin type 3 from London somewhere, okay? So not a highly drifted strain, but it was there, probably imported from an area where they've been immunized. And from Pakistan, there was a CDVP2, which is not, again, unusual, I don't think, not to be unexpected. We haven't turned up many nasties here. The issue arises what happens if you do turn up a nasty, okay? And the first thing you do is not panic, okay? Because your current immunization schedules are clearly good enough to control it because you have no polio in this country. So you can actually afford to take a bit of time to decide what you're actually going to do about it before you actually start launching mass vaccination campaigns left, right, and center. Okay? I don't think this is a risky proposition. There are others who disagreed, I should say. So what do you do about oral polio vaccine and the problems which are associated with it? And the answer is you stop using it in big black writing. VAPS, clearly vaccine-associated polio won't happen if you're not using oral polio vaccine. Okay, so stop using it. And we did that in 2004. CVDPVs, stop using OPV again. Okay? You can get rid of the CVDPVs by use of monovalent oral polio vaccine, which is what happened in Nigeria, where they had a lot of type 2 CVDPVs. Okay? And the argument is that it doesn't happen every time you do this. It's not a 100% event that you get a CVD-PV, but you may get them anyway. I mean, it can happen. It's not 100%. So you can get rid of them by doing that. IVD-PVs, it's not clear what you do with that. We tried a number of treatments on this guy in where he came from, and some of them were quite amusing. He was given assorted oral antivirals, which did nothing. He was given oral immunoglobulin. He's a vegan, so he doesn't like to mix it up with milk, but he doesn't mind drinking human blood. I think that's funny, but nobody else does. We also had a local breast milk bank. Okay, so he got a load of that for a couple of weeks on his cornflakes. It might have done something, but we're not totally clear whether it did or not. Okay, none of the treatments really stopped it. Okay, as soon as it stopped, it came back again. There are efforts to develop antivirals against polio, and if there was an antiviral drug, I think that would really be quite useful. It's not necessarily a commercial proposition, but it would be quite useful for treating these kind of guys, and also if you have outbreaks, maybe that would be a reasonable way to actually bring them to a halt if you used it widely enough. And finally, there's the AVD-PVs. Well, again, you stop using the oral polio vaccine, but as you don't know where they come from, who they're from, it's almost impossible to know what happens next. Okay, so the rest is slightly tricky. What's happening uniformly throughout the world now is that countries are moving independently to the use of inactivated polio vaccine. So we switched in 2004. Europe does not use oral polio vaccine anymore. Okay, it's just swept across it. America and Canada use IPV and not OPV, which they used to use, as we did. And Mexico is changing, Argentina's changing. It's gradually going in that direction. It's going through Europe, to America, down through South America, and then who knows what. Okay? So there's a definite trend to actually do that, to get rid of using OPV. What about stopping vaccination? Because, as I have said in past occasions, if your last case of polio is in 2020, it's inconceivable that you're still immunising against it in 3020. It just doesn't seem sensible. So how do you stop it? So one of the things that they're starting off with is withdrawing the type 2 component, because you haven't had a type 2 wild type since 1999. And most of your VAPPs that you see now are caused by the type 2 component. So there you are. Most CVD-PVs are type 2. Most IVD-PVs are type 2. And not only that, the type 2 component is the most effective of the OPV strains. So they only really got rid of it in India when they dropped it. When they only got rid of type 1 and type 3 when they dropped it, because this one competed off all the other two. Okay? So it's far too effective. It causes all sorts of problems, and there's no disease to protect against. The only reason you're giving OPV with type 2 in it is to protect against the OPV with type 2 in it that you're giving. Okay? That, again, doesn't seem logical to me. Right, so how do you withdraw it? The first thing is that there are routine uses of vaccine and there are campaign uses of vaccine, and you have to withdraw it from both at once. And I think right at the very beginning they were talking about just withdrawing it from the campaign uses, and that really is a recipe for disaster, isn't it? Because you carry on seeding it in the routine immunisation programs into a population which is only half immunised, if you like. That's a guaranteed way of getting a CVD-PV out of it. You can eliminate the CVD-PV that you know about, either with a trivalent oral polio vaccine or with a monovalent type 2, which is what they did in Nigeria. If there is an outbreak of a type 2 later on, after eradication, you must have something in the cabinet to actually wheel out and deal with. So you have a stock of monovalent OPV to go. And WHO are also saying you need a single dose of IPV to protect people against disease, just in case there is a type 2 around. And in that case, the only source of type 2 will be laboratory or manufacturing sites, and containment of all type 2, whether wild or type vaccine, is really needed. And that was actually done this year. So if you have a type 2 poliovirus in your lab and you're splashing it around on the bench, you shouldn't be. It's recategorized to type 3, okay, and it's not an easy thing to deal with, which I'll talk about in a moment, because there are serious implications of this. The view is really that this is an experiment, if you like. I mean, that it may well be that type 2 has been eradicated. It may be that it will re-emerge. And if it re-emerges, then they will all be braced to do something about it, in theory. But it will also come for type 1 and type 3 in due course. The implications are here. First, the IPV supply and safe manufacture. Currently, most manufacturers in Europe, Sanofi make an awful lot of it, GSK make an awful lot of it. Okay? I can't really believe that countries like China and India will actually wear that for long, because I think they are perfectly capable of making this stuff, but it needs to be done properly. Okay. The supply at the moment is insufficient, okay, and so the idea that everybody gets one dose of IPV is just not holding water. You cannot do it. You've got countries which are prioritised in terms of who gets what. IPV is expensive to produce, and it uses wild-type polio. So we know that these polios will cause poliomyelitis because of various accidents in the past. So it might be an issue where you've got a country that's never made this stuff before, okay, and where maybe the infrastructure is not as wonderful as you want it to be. So why not, says WHO, make it with the safer Sabin strains, okay, which are attenuated and which you've used in the eradication program. Now, the fact is, of course, that you know that the Sabin strains aren't 100% either, because they cause all these other problems, if you like. So in any event, in my view, this would be a short-term fix, okay, if it's a fix at all. And the other issue, of course, is that the quality issues, which is what NIBSC loves to deal with, are absolutely horrific. I mean, Sabin strains are different in their immunogenicity. Determining what you've actually got there is going to be very different. You're going to have to do clinical trials of combination vaccines. Manufacturers in Europe do not want to do this at any price whatsoever. And I can sympathise with that. Finally, you must have your polio contained. And the model here is, again, smallpox. And I think it's a mistake. Okay. I shouldn't really be saying that in a public forum. I think it's not quite as well informed as it might be. Okay. Polio is not smallpox. You can synthesize a polio genome in 2 weeks and have the virus in your hand in 3. So synthetic biology, I was impressed to actually think that synthetic biology actually impacts on the polio eradication program when I was listening to this earlier, actually. So the areas where you clearly do need it are production units. The amount of virus which is put into IPV production is absolutely gigantic. You get 10 to the 12 PFU per mil, and seals do fail, and workers tend to get a face full of this stuff, okay? And then what do you do, okay? So production units must be contained. You cannot just dump this stuff in the local river and hope for the best. There are virology diagnostic labs which may have access to polio specimens or may be looking for polio specimens in particular. You've got virology research labs who may be persuaded to actually stop working on polio and work on something else, and I think that's been quite successful, in fact, okay? And then there are labs which may have a specimen with polio in it and they don't know about it. So if you, for example, were looking at intestinal parasites in Nigeria over the last 10 years, you've probably got fecal specimens that you may have kept, okay? And they could well have polio in them, okay? So it's a risk, right? Finally, there's any lab that might have a full-length genomic polio RNA as it's infectious, okay? And that actually gets down to the point that because you can synthesize this in 2 weeks, you're almost saying any molecular biology lab should actually be working to GAP3. And it's just not practical, I don't think. So the first two, I think, are clearly major priorities. The rest of it, I'm not so sure about. And it's a very, very serious document, this. It's a very, very heavy document. There used to be 55 odd labs in this country who had polio, and there are now about 3 or 4. Okay, so I think they've been persuaded to stop doing it. And I think that's for the good, actually. I'm not sure that's a bad thing at all. So what can be done? GAP3, I think, needs a bit of thinking about, okay, in terms of how you apply it and what you apply it to. Because you can't say molecular biology is going to stop because we might get someone making a polio out of it. And it's not difficult to do it. It really isn't. So one of the things that you could think about is the major risk areas, which are production of vaccines and using them in lab use, like for serological assays and things like that. So one of the things that you could consider is strains that do not revert or transmit. And you could also think of strains that do not grow in people, but grow in culture, okay? If you can design them, which of course, as I'm saying this, we can, okay? And finally, you could consider empty capsids or replicons, okay? So just things which are just got no replicating RNA in them at all, okay? Or you can carry on and hope to get away with it, okay? And I think we shall see which one goes the way it goes, okay? What I want to do now is talk about 2 things which happened at NIBSC, where we're looking at safer strains for production and for use in the lab, if you like. So these are determinants of attenuation in type 3. So this is the Sabin type 3 strain, which does not paralyze monkeys. If you take these 2 mutations out and revert them, it will paralyze monkeys, okay? So these are the things which actually knock it on the head. And there are 2. There is one in VP3, which is one of the capsid proteins. Now, the way the capsid is put together, you get 5 units of VP1, 2, 3, and 4, and they come together to form the pentamer. You then get 12 of those get together and form the icosahedral capsid. This particular mutation lies at the precise interface between the protomers that make up the pentamer, and they make the pentamer unstable. Okay? So that's the reason why this attenuates, that the vaccine just basically is not able to grow very well at high temperatures because it's less stable. The other one is the 5' non-coding region. So polio has a particular strategy of replication, and the 5' non-coding region is an iris, or an internal ribosomal entry site, for reasons which we won't go into. This particular structure here is called domain 5, okay? And each of the Sabin strains has a mutation within that region which make it weaker, okay? So you destabilize this, and what happens is that you basically screw up the ability of the virus to translate its protein, or to initiate translation of its protein, okay? These things are for real, because when you put these vaccines into babies, my first children were the first experimental animals on which this was done, okay? What you find is that they revert. So the U that you see down there, the U with the 3 in the brackets, that's the mutation in type 3, okay? And that reverts within about 48 hours in our hands anyway, or in my son's bowels, I should say, okay? So what that means is that probably a U at that position is incompatible with replication in the gut, okay? It just goes in one end, changes and comes out the other, okay? And the same thing with the other. The type 1 has a GU there, and the type 2 has another one over the other place. And the whole thing is very consistent with the idea that you're weakening that stem structure there, and that actually has a major effect. So what happens if you weaken it to death, if you like? And that's what's been done here. This is Andrew McAdams' work. And what's been done here is to the Sabin 3 strain is up on the left-hand side, and you see there's a UG next to that 470. So one thing you can do is make that a UA. That means that you need 2 simultaneous mutations to revert that back to the wild type, okay? It's a bit stronger than a GU, but it won't go back to a GC overnight very easily. You can then take the other GCs there, and you can change those to UAs as well. So you're weakening the structure as you go along. And S15 is very similar in its neurovirulence and in its growth properties to the Sabin 3 strain, okay? So it actually sort of works, I think. S17 has taken that a bit further, swapped a few more GCs around. S18 has swapped more of them. S19 has more or less copped the lot, okay? S19 is a fairly crippled up virus, okay? It doesn't really do very much at 35 degrees. It'll do stuff at 33 degrees, and you can actually get satisfactory quantities of virus coming out of it. If you put it into monkeys, the monkey is not a very good oral model, if you like, of infection, but it doesn't infect them, okay? And these things are very stable in culture, and I believe that they really, really will not actually infect anybody. You cannot put enough of that into an animal to actually get any kind of paralysis at all, okay? This, I think, is a safe strain for actually use in production, okay? Provided you treat it properly, okay? And you could also use it in serological tests as well, I think, without much containment at all. And we are getting a risk assessment together to go to HSC to see if they agree, okay? If they do, we'll see what happens. The other thing, which was just published in PLOS Pathogens this week, is thermostable empty capsids, okay? So this is not necessarily a crystal clear slide, but if you can see that pale blue blob up in the top left-hand corner there, that is the VP3 91 change that makes things unstable, okay? A protemer has been kicked out of there so you can see where it lies. The white dots are the ones where you see revertance in vaccinees, okay? So they don't tend to change the phenylalanine back to a serine, which is what it is in the wild type. They put in second site mutations to compensate for it. And the argument, of course, is that the blue change makes it unstable, the white changes make it stable, right? So that's a way of stabilizing your capsid if you put those in anyway and you see what goes on. And there are a number of those which are there, okay? And the result of that has been that you can produce empty capsids, which are normally so unstable that you just snap your fingers and they fall to bits. You can produce empty capsids which are stable as inactivated polio vaccine, and you can store them at 37 degrees for 6 months without losing very much. I think they're magic, actually. I think the production platform is maybe a different issue, but I think they're wonderful, and you can apply this somewhere else, I'm sure. Right, so this is my second to last slide. Wild-type polio, I think, is very near the end. Okay, we're at phase 3.5 here. Okay, with 40 cases, I don't see why it shouldn't be gone in a couple of years. That's a very dangerous prediction to make because everybody's made it before and they've been totally wrong from 2000 onwards. But it just looks like it's on the way out somehow. Type 2 OPV has been withdrawn from regular use, and this is an experiment, okay, which they can reverse if they so wish, if they need to. There are major issues of containment, and they are a big current concern and spread well beyond polio. For example, if you consider the synthetic biology discussions we've been having today, this actually plays into that. This is synthetic biology in real life. One of the other polio strains which has been talked about has been a codon de-optimized polio. You can make the thing in 2 weeks and get it in your hand in 3. Okay. So it's an issue, and it's being dealt with basically in the polio area. There are acceptable safer strains or new platforms. Maybe they could help in production and in surveillance in clinical diagnostic labs. We're very, very near the end of this. Okay. And this is my last slide. Okay. Thank you.